Welcome to the BioEduQuest Saif YouTube channel, your ultimate destination for all things biology. Join me on an exciting journey of exploring the wonders of life, from the tiniest organisms to the complexities of ecosystems. Transcription of DNA and Central Dogma. The discovery of DNA was one of the most significant findings in the field of science. Today, deeper insight into the DNA structure has unraveled answers to questions that have been around for eons. The Central Dogma explains how the DNA codes for the proteins which proceed in three stages, namely, replication, transcription and translation. Once DNA replicates its two strands, the information is copied into RNA by the process called transcription. Let's learn more about the process of transcription. Transcription definition. Transcription is the first step of gene expression that involves the formation of RNA molecules from DNA. What is transcription? It is one of the first processes in gene expression. The genetic information flows from DNA to protein, and this flow of information takes place in a sequential process of transcription and translation. Only one strand of DNA is copied during the process of transcription, known as the template strand, and the RNA synthesized is called the mRNA. The main motive of transcription is RNA synthesis from the DNA sequence. The RNA transcript carries the information used to encode a protein. RNA polymerase. The RNA polymerase is the main enzyme involved in transcription. It uses single-strand DNA to synthesize a complementary RNA strand. The DNA-dependent RNA polymerase binds to the promoter and catalyzes the polymerization in the 5' to 3' direction on the template strand. Once it reaches the terminator sequence, the process terminates and the newly synthesized RNA strand is released. Transcription unit is a stretch of a DNA transcribed into an RNA molecule. Its function is to encode at least one gene. Suppose if gene encodes protein, then mRNA is produced by transcription. A protein encoded by the DNA transcription unit may comprise a coding sequence. Compared to DNA replication, transcription has a lower copying fidelity. Stages of transcription. Transcription proceeds in enzymatically catalyzed steps, i.e. 1. Initiation. 2. Elongation. 3. Termination. Initiation. RNA polymerase attaches to the DNA molecule and moves along the DNA strand until it recognizes a promoter sequence. These are known as the transcription start sites. The DNA double helix then unwinds and all the bases on each of the DNA strands are exposed. This acts as a template for a new mRNA strand. Elongation. Ribonucleotides are added to the template strand that enables the growth of mRNA growth. Termination. RNA polymerase encounters a terminator sequence and the transcription stops. RNA polymerase then releases the DNA template. RNA processing. The transcribed RNA is known as the pre-mRNA. It is processed further to convert it into mature RNA. RNA processing include 1. Capping. 2. Polyadenylation. 3. Splicing. Capping. A methylated guanine cap is added to protect the mRNA. It involves 1. Addition of methylated guanine. 2. It occurs at 5' end of mRNA transcript. 3. It protects the mRNA from degradation. Polyadenylation. The poly tail also protects the mRNA from degradation. It involves 1. The endonucleases cleave the mRNA at a specific sequence. 2. The enzyme polymerase facilitates the addition of several adenine nucleotides. Splicing. 1. The non-coding sequences, i.e., the introns, are removed by splice-osome excision. 2. The coding sequences or the exons join together by ligation. Thus several proteins can be made from a single pre-mRNA. A mature mRNA is obtained at the end of transcription. Frequently asked questions. What is the process of transcription? Transcription is the process in which a DNA sequence is transcribed into an RNA molecule with the help of enzyme RNA polymerase. One of the DNA strands acts as a template to make a complementary RNA strand. Where the transcription starts and terminate? The transcription starts at the 5' end of the DNA sequence. Are enhancers necessary for transcription? Enhancers are regions in eukaryotic cells that help to increase the transcription. These are not necessarily close to the genes they enhance. What is the end product of transcription? An RNA transcript is obtained as an end product of transcription. It can form any type of RNA such as rRNA, mRNA, non-coding RNA and tRNA. The prokaryotes form a polycitronic mRNA, whereas eukaryotes form a monocitronic mRNA. What are the promoter sequences? Promoter sequences are the gene sequences where the DNA transcription begins. These are located upstream at the 5' end of the DNA sequence. BioEduQuest Saif YouTube channel, your ultimate destination for all things biology. Join me on an exciting journey of exploring the wonders of life, from the tiniest organisms to the complexities of ecosystems. In conclusion, the journey through the world of DNA transcription and the Central Dogma has illuminated the incredible complexity and precision that underlies the expression of genes. From the discovery of DNA to the intricate process of transcription, we've explored how nature encodes information for life itself. Transcription, the first step in gene expression, showcases the role of RNA polymerase and the transformation of DNA into RNA. This RNA, in its various forms, holds the key to building the proteins that define life's functions. As we've seen, the process of transcription involves initiation, elongation, and termination, each step finely tuned to ensure accuracy. Post-transcription, RNA undergoes essential processing steps like capping, polyadenylation, and splicing, turning it into mature RNA ready for translation. So, whether you're captivated by the elegance of molecular biology or simply curious about the science of life, understanding transcription is a fundamental step towards unraveling the mysteries of our genetic code. With every new discovery, we inch closer to understanding the intricate dance of molecules that govern life as we know it. The journey continues, and with it, the promise of new insights and breakthroughs in the endlessly fascinating world of genetics. Thank you for joining us on this exploration of DNA and the Central Dogma.